Okay, our next speaker is Daniel Toloudis, a principal engineer and associate director of software engineering. You have quite the title at the Allen Institute for Cell Science. His path to the Allen involved a career involving many interesting uses of 3D graphics, for which he has a slide for, so I won't even attempt to go into it in more detail. And also, happy to see another Hopkins alum, his Bachelor in Physics and Master of Engineering from, or Master's of Physics from University of Oregon.
But be careful if you do. The yellow one is forward.
So yeah, my name is Dan, and I'm a software engineer at the Allen Institute for Cell Science, and I'm gonna go a little bit high level and give a little background first, and then talk about the history of how we brought Zarr into the Institute, and then where we're gonna go from here. So a little background about me first, just for fun. I joined the Allen Institute in 2016 when it was still in early stages of the Cell Science Institute. And before that, I had this whole other career developing 3D rendering software. So I worked in a variety of different industries, from office to furniture, planning, layout, to product marketing, to animation for actual like entertainment movies. And I wrote the code that rendered all of these images. So now I work on similar types of things, but focus on cell science. There's an animated cell playing here. I would have to request access. I'm not going to. The important thing here is that at the time that I joined Cell Science Institute, our mission statement had two parts to it. And the first part is to understand how cells organize themselves and change. The second part is more closely associated with the teams that I work on, developing and democratizing tools. So what that means is we build software tools that help make our scientists' lives better, make their work easier or faster, and then tools that also can be used to bring the results of the Institute to the outside. To make our data more interesting, more accessible, more easy to use. So the research of the Institute, which is broadly encompassed by this set of pictures, when we think about this internally, we can sort of start to categorize it into these three areas: visual analysis, image analysis, and data analysis. I'm characterizing the tools that are used to do some of this research. So we can define these categories as visual analysis being human-driven exploration and interpretation, image analysis as in image transformations or extracting numbers out of images, and data analysis meaning statistical and computational analysis of quantitative data. So we can try to like fit tools into these categories, but most of the time there is some cross-pollination between them. So like, it's a convenient way to think about it, but a lot of the times the tools come up. But we can categorize many of the solutions we've built into these areas. And then there are some foundational tools that we've also built that kind of reach into all of them at the same time. BioIO is one of them, which is our Python library for reading and writing image data. And BioFile Finder is our front end for making data sets more accessible. So, okay. Our journey begins back in 2016, 2017, when Cell Science was still a new institute. Our early goal included doing this automated imaging of thousands of 3D fluorescence microscopy images from our gene-edited cells. We knew we wanted to, we're an open science and open data institute, so we knew we wanted to release these large image data sets publicly. And early on, it was clear that the open format of choice was OME-TIFF. So in those early days, among the first tools we developed were this image loading library called AICS ImageIO at that time, which now is known as BioIO. And we also built a web-based volume viewer because we knew we were going to be presenting our data. And it was, because it's all 3D, we wanted to really emphasize that with the volumetric viewer. So early on, it was obvious that all the problems with our data are the problems that Zarr solves. Large monolithic files that can be unwieldy. It's difficult to share these files, to move them around, to visualize them. In particular, our web volume viewer, because we wanted to make it web accessible, we actually downsampled these images into a completely different sort of ad hoc format wedged into PNG files that then download into our web viewer. So moving ahead to 2019, this is the first instances of the Allen Institute for Cell Science becoming aware of Zarr. If you can read the bottom of the slide, this is from our Slack messaging history. What is this Zarr thing? I don't know, another large file format. Yeah, it looks like chunked HTML5. So that was like the very first mention, and for a long time, nothing happened. And then we kept talking about it and building up momentum. And then finally, in 2021, a plan occurred. Yeah, so this is sort of the instance of developer takes a cup of coffee and goes into a cave and comes out 4 days later with a prototype. So the strategy was, let's just make a Zarr and see if we can make our viewer loading. So that was the very beginning. It turns out that having a working prototype that you can show people is really, really powerful, even if it's just barely working. And it was immediately obvious benefits of using Zarr. In particular, multi-resolution, right? And once we had this data loading in our web viewer, it was also clear that now suddenly we can load the same file format that's basically where the raw data lives in our web-based viewer. We can just drop it in the cloud and access it from there forever. Yeah, and multi-resolution is really important for your active viewer. Okay, so from then, we progressed to develop these tools. So now we have this working proof of concept. Now we have to make it production ready. So on the visualization side, the volume viewer, which has now been named Volley as of not too long ago, has had a lot of work done to handle multi-resolution levels better, handle time series data. It has an in-memory chunk cache. So it's kind of beefed up its robustness. We used to call this our website 3D cell viewer. But as soon as we added Zarr capability, now it can basically load any Zarr. So it's not just a cell viewer anymore. We also have this desktop viewer called Agave, which is basically another volume viewer, but it kind of unlocks all the limitations that you would have in the web browser and lets you just throw much larger data at it and crank it on the GPU and churn out much nicer quality renders. And we used the TensorStore. This is a C++ application. So we folded TensorStore in to support Zarr. TensorStore is a great library, and it was really easy to integrate it. Meanwhile, we had to build this capability into our Python library, BioIO. So over that time, the ICS ImageIO was restructured and renamed to BioIO. It still has the same API. We added Zarr support for reading and writing. We started converting some of our data sets, and we actually publicly released our first OME-Zarr data set during this time. And at the same time, we also started beefing up some of our conversion capabilities. So conversion is difficult, and BioIO just does the lowest level parts of that. So we needed to build a slightly higher level library to manage some of our much larger file conversions. Also during this time, we contributed to this OME-Zarr publication that talks about the breadth of tool creation around OME-Zarr. We gave a variety of internal talks advocating and showing off demos. Also, interestingly, we added Zarr support to our centralized file management system, which was kind of a heavy lift because this system used to be sort of like one entity per one file. And handling multi-file files was and then at the same time, or shortly after that, we developed this file finder tool, which originally was developed as a front end on top of our centralized file management system, but we generalized it to be able to load data sets from basically any backend. And yeah, I can link out to our viewers. So, oh yeah, and because we think File Finder is important and a really useful tool, we have quick pitch for talks tomorrow that are focused on File Finder. I'll show them those. Okay, so that basically brings us up to the present. Very recently, we've added Zarr v3 support, OME-Zarr 0.5 spec, to be able to load them in Volley and Agave. We have just barely started work on adding Zarr v3 support to BioIO. This is like in very, very early stages, but we will have it done this year. And we also just started seeing adoption into our scientific workflow. So there was like one very early, like naive result that shows an immediate speed up just for switching from TIP to Zarr. Pretty naive choices of chunking. So that's the present now. Sure, anybody who is thinking about how to deal with Zarr data is thinking about all of these things and maybe some others. But we have some basic thoughts, like these are definitely areas where we need to put work into moving forward. So format conversion is still, format conversion is really slow. It's a pain. Our code is in this sort of barely working state where we can do it, but it really has a lot of room to optimize. Some of the things we're thinking about are how soon after image acquisition can we actually do conversions? Today, we don't have a good system for doing it early. We collect a lot of data and then convert large batches. Basically, when we're ready to release. Writing directly to cloud would also be a great optimization for us. We have limitations with our internal file management system where we need to write to on-prem storage first, then copy up to cloud. So that's an area we could improve on. Also, we are pretty sure we're not maxing out our parallelism with these conversions. We are doing some things, we're using Dask, but we're not completely fully optimized. I think we have a lot to improve there. And we haven't even started thinking about rewriting into like subsets of a Zarr. We basically, if your Zarr failed somehow, you're deleting the whole thing. Oh, there we go. Okay, so let me make sure. Yeah, okay. So for chunking, our initial choice of chunking was made very early on and quick, and it has worked out well for us because it was focused on visualization. So we haven't really done a lot of exploration with chunking strategies yet. Our first choice was, let's pick an integer number of Z slices that fit in a fixed download size per channel. So our viewer is optimized to show multi-channel data. We selected 16 megabytes as a reasonable size that the user would be willing to wait for some data to load out of their browser. And so this choice allowed really fast single slice viewing, and it also allowed interactive 3D viewing up, downsampled resolution. So it will show higher res single slice photos. But of course, this may not be optimal for certain types of ML training where you want box-shaped chunks, or certain other types of applications. If you know your whole volume data fits in memory and the latency of the first access isn't as important, you might choose really big chunks that fit the whole volume. So this is really, really sensitive to use case and access patterns. Okay, so I'm going to run through a quick sort of back of the envelope app example to show some numbers. A typical image that we capture might be about 2000 by 1500 by 100 at 16 bits per pixel. This is rounded up a little bit from what we actually capture, but it's close. So if we use our 16 megabyte chunking strategy, we can fit 2 slices of this Z stack in memory, or sorry, in a chunk. when we downsample, because our Z resolution is so much less than our XY, we only downsample XY by half. So at that next level, we can fit 10 slices in one 16 megabyte chunk. Also, I should say, all of these calculations are based on uncompressed data. Compression only makes this better. I'm just doing worst case, naive choices. Okay, so then we can downsample again one more time, and now our 116 megabyte chunk covers 42 slices. So yeah, again, compression makes this better. So now let's see how many files we're generating this. For our typical data with Zarr v2, our top level original data can be stored in 50 files, 16 megabyte chunk size. When we add in the multi-scales, you do a little bit of integer math with rounding, and you get 63 files for the example. Two multi-resolution levels. But the problem is we capture multiple channels and multiple times. So we captured 3 channels of data at 200 times. Data. And we're processing a data set now that has like 1,000 of these time series in it. So we're already up to one time series has 37,800 files. This is obviously a lot. We're getting into big numbers now. So if we look at sharding, and we just randomly pick a decently large shard size, I don't even know if this is large or not. Say 256 megabytes. That means it can hold 16 of our 16 megabyte chunks. If we naively divide by 16, we get this number, but that number is not quite accurate because we still have to separate each of our multi-scales. So the true number that you get, you actually do this integer math, you get, we go from 37,800 files down to 3,600. So it's a pretty good increase for this arbitrary selection of shard size. So there's a lot of experimentation to do on like optimizing this. So even though none of my calculations accounted for compression, we do compression. We just currently, I think we just use the default, which I think is BLOSC. I'm sure. This has not really been a big consideration for us yet. It's really, the total file count is much worse of a problem for us than compression concerns yet. It's just too hard to move these things around right now. So probably for us, optimizing for compression is a to-do, which we will combine with our choices of chunking and sharding. So we have a bunch of work to do to like experiment with different parameters here. Yeah, and it's just interesting to note that segmentation data compresses so nicely, like just it's dramatically better than Raj data. Okay, so yeah, I couldn't resist. So for metadata, the direction that we've seen from the last conversations that I had about OME-Zarr is that it seems to be heading toward a notion of decoupling from metadata. We think this is totally reasonable. You know, we've been designing systems like BFF to basically do that very thing. One of the interesting future avenues for BFF is, so currently BFF stores metadata in like a really flat, just name value pair list. One interesting feature investing is more structured metadata. And also because BFF can load arbitrary metadata sets and bring them together, clearly for heterogeneous data sets, it would be really interesting to think about how to distinct this. Okay, so collections are a big, interesting concern for us. So in terms of when we think about tool support for collections, you can immediately go to Zarr groups, but we wanted to commit to a data organization strategy that puts things in Zarr groups. We think they're too tightly coupled. Basically, for us, individual independent Zarrs are much more flexible for the moment. So we've added support to Volley. Volley can now load multiple different Zarr URLs and combine them as separate channels with the same volume. And it can also load independent Zarr URLs and treat them as completely independent images that you can page through. And the way we did this was just with the absolute simplest possible spec for a collection. We just built like really simple lists of URLs with metadata. Yeah, so I think there isn't really a good collection spec yet. We would like to get to one. Another interesting direction that we are thinking about a lot is file provenance, where you can build collections from linked files where maybe the files are, segmentation knows that it's derived from. So that kind of hierarchical information will be in our database. We build collections out of that. So yeah, in terms of use cases for collections, so clearly plates and wells is a very standard thing. We know that's already in the NGFF spec. Again, for flexibility reasons and not wanting to commit to a particular data organization method, we still store all that stuff in independent ZARs. We just have separate metadata for our well and plate. Another interesting use case for collections. Oh, okay, I'm actually near 30 seconds. So coupling raw images to segmentations, obviously for a collection, but most interestingly, public data sets, really large collections, right? And selections of subsets of data sets. So we have this really interesting future use case for BFF where we can make selections of data and then forward those collections to other tools, whether it's a visualization tool or even just a cloud compute from workloads from BFF. And if we can do that with ZARR as the backbone of this whole thing, then we can come to a sort of a unified, standardized experimental workflow. All of our experiments get standardized to OME-Zarr early with our sort of nearly schemaless metadata and all analysis and processing can flow from that. And that is my last slide. These are the key contributors who helped implement ZARR support in all our tools. All of our stuff is open source. There are the links.
I just want to comment that we should talk about collections. We had a fair amount of discussion about that at the NGFF hackathon last year.
Maybe it's here.
Okay, questions? We have a couple of minutes.
Thanks for the great talk and thanks for all the work, especially. As part of your metric, because yesterday Jason mentioned the work we've done with people in Indonesia and the work that Swish is pushing in terms of Asia. Sometimes obviously the network bandwidth can be a problem. Did you consider that part of your review? Like, you mentioned performance of and the various chunking strategy, etc. When the bandwidth cannot be as good as what we will expect depending on the country you are in.
I think we're not network bandwidth limited internally, but we are network bandwidth limited for shipping data out to the cloud.
Yeah.
And that's part of our parallelism optimization workflow. I think the way that we currently send data out is still not optimal. So we definitely.
Okay.
Thanks.
I have time for another question.
Okay, I'll be provocative. This is great. It's so amazing. Let's just take the logic to the limit, the full decoupling of the binary data and the metadata. BFF is so powerful and where that's going, but then you have all of the tooling that has to support whatever metadata representation you have. So you're signing up for that? Sorry, I said, I say this as the old guy in the audience, right? 25 years later.
When you say all of the tooling. Sure.
No, it's great. It's just.
I mean, I think right now the beauty of BFF is the simplicity of the.
Yeah.
It's very, very.
I guess what I mean is, okay, so the promise is that anyone can find these files and then work with them.
Right, so there is a farther vision of the Allen Institute. So this is the everything box here.
Yeah.
Right, so there's a farther reaching vision where we can describe that as cloud accessible computational workflows. With BFF front, where we let people either rerun our analysis workflows on our data or bring their own data to it.
Cool. Thank you very much. Cool. Our next speaker is Stefan Zellfeld, a group leader at Genelia. I had the pleasure of first meeting him in 2011. A cat-made hackathon, a web-based toolkit for annotating data. Since then, he has used his wizarding skills for good. He's collaborated with many labs and projects and has a particular niche for those with absurdly large and often ugly data that needs some taming. So yeah, here's Stefan.